Assalamualaikum warahmatullahi wabarakatuh and good afternoon to the fellow audience. We are STP student from Group 10. We'll be presenting about our project titled Green Adsorbent for Water Filtration System. Our group consists of 4 members, me, Nur Izzah, Adam, Madeline Teo and Melanie Rachel, and has been supervised by AP Dr. Cecilia and Mr. Faisal. First of all, let me briefly explain about the introduction of our project. As for the problem statement, we have identified that heavy metal pollution in water sources has been a major problem over the centuries. The presence of heavy metal in rivers, particularly the lead, aka the plumbum, and copper, severely may affect the water quality and eventually imposes detrimental effects on human health. As reported by healthychildren.org in 2021, high levels of lead in tap water can cause health effects if the lead in the water enters the blood and causes high blood lead level. It can cause damage to the brain and kidneys and can interfere with the production of red blood cells that carry the oxygen to all parts of the body. While for copper, there is actually a disease known as Wilson disease, and as reported by AgriLife Extension in 2021, people that affected with Wilson's disease, a rare genetic disorder that affects approximately 1 in 30,000 people worldwide, cannot excrete excess copper. Copper can accumulate in these individuals to dangerous levels, and if not detected and treated, it can cause death. So the solution that we are bringing in this project is by introducing water filtration system that utilize the deabsorption that can improve the water quality by looking into the extraction of heavy metals, particularly for lead and copper. In this project, we have constructed a water filtration system. There were some layers of sediment filters with 4 different sizes of sand and a layer of activated carbon impregnated with ionic liquid. In this project, we are comparing the absorption efficiency of heavy metal, which are the lead and copper, by 4 different adsorbents with 4 different loading of ionic liquid in the activated carbon. And for our project, we have selected Sungai Kinta, a river in Perak, Malaysia, as our sampling site, which is the source of our water sample. The location of our sampling site is at the Kinta Batu Gajah River Bridge, which is very near to the urbanized area. As emphasized by Wakib et al. In 2020, Sungai Kinta is one of the most polluted rivers in Perak due to its location near the developing industrial and agricultural area. Next, the objectives. There are 4 main objectives in this project. First is to produce an ionic liquid that is suitable to be incorporated with activated carbon. Second, to identify the appropriate parameters that is suited to filter contaminants. Third, to analyze the absorption efficiency of heavy metal in the produced water filtration system. And fourth is to implement the green aspect in the water filter, such as the recyclability and the safety.
We will now move on to the literature review or data gathering segment of our project. Activated carbon is readily an effective and popular adsorbent for a wide range of organic and inorganic pollutants in aqueous and gaseous mediums due to its well-developed internal microporosity and broad range of surface functional groups. Activated carbon is also easily obtained as it can be made from high carbon content trash in the environment such as coal materials and rice husks, coconut coil or fiber. Also, due to its large surface area which ranges from 500 to 1,500 meters squared per gram, equipped with its microporous capabilities, and chemical complexity of its surface area, activated carbon has a high potential for absorbing heavy metals. Research on activated carbon modification is also gaining traction in recent trends to create activated carbon affinity for specific contaminants in the removal from water. For example, absorption of imidazolium-based ionic liquids onto activated carbon has reported effects of cations and ions and surface chemistry on absorption efficiency. Besides, increasing length of active chain and the anion or the hydrophobicity of the cation increases its absorption capacity. Hydrophilic ionic liquids can also adsorb on an activated carbon surface with hydrogen bond donor groups. Therefore, ionic liquids with longer structures will have higher capacity for adsorption on activated carbon.
Moving on to the next part, I will share about the methodology of this project. So first thing first, I will share our design concept. So we have drawn a design for our water filtration system where we have combined the chemical and sediment filters into one filtration system. So the reason why we choose these 2 filters is to increase the effectiveness of the filtration. The chemical filters used for this project was the activated carbon, where it is responsible to remove any dissolved chemicals in the sample that will pass through the system later. While the sediment filters was responsible to filter out any visible impurities like maybe dust or debris. So for the arrangement of the layers, which is the filtration unit, we have been arranging the layers of sediment, which are very coarse sand, medium coarse sand, and also fine sand by referring to the Wentworth scale. So the layers are arranged based on the size, where the larger size will be on the top and the smaller size will be on the bottom. So the activated carbon was put in the middle of medium coarse sand and also fine sand because the size of the activated carbon was in between the size of the medium coarse sand and also the fine sand. And lastly, we end our layers, the filtration units, with a layers of cotton in order to prevent the leakage of the sediment and activated carbon into the filtered sample later. Next, we have also prepared the sediment filter by sieving about 2 kg of sand using the sieving machine in order to separate the sand into various grain size. The sand of various grain size collected was very coarse sand, medium coarse sand, and also fine sands with the range size of 1.18 mm to 115 micrometer. So the next material that we prepared was the activated carbon. So we have been using about 200 grams of activated carbon. So the activated carbon was first heated up to 110 degrees Celsius in a vacuum oven in order to remove water traces from it. It is then put in a desiccator to prevent any moist accumulation in the activated carbon during the cooling method.
Okay, so to prepare the ionic liquid, we need to synthesize 1-butyl 3-methylimidazolium chloride, which is the BMIMCl, and sodium dicinamide, which is the NaDCN, to produce 1-butyl 3-methylimidazolium dicinamide. By using metathesis reaction, we will obtain the sodium chloride, which is the salt, as the byproduct. Based on the stoichiometric calculation, 1 mole of BMIMCl will react with 1 mole of NaDCN to produce 1 mole of BMIMDCN. So we need 50 grams of the product to be used in this experiment. So we need to calculate the mole of BMIMDCN, which is by dividing mass with the molar mass. So we'll get 0.0731 mole. As 1 mole of both reactants, we will produce 1 mole of the product. So the mole of both reactants will be the same, which is 0.0731 mole. So we need to calculate the mass of both reactants. So for BMMCl, we need to multiply the mole with the molar mass, so we'll get 12.77 gram. While for NaDCN, we will get 6.509 gram. So after calculating both reactants, we need to mix both of them to be dissolved in 50 milliliter of acetone. Then it will be stirred for 20 hours using a magnetic stirrer at room temperature. After that, it will be sealed in round bottle flask using paraffin film, and it will be kept at negative 20 degrees Celsius for 5 days. Then the mixture will be filtered using filter paper, and it will be evaporated using a rotary evaporator. It will produce a colorless ionic liquid instead of slight yellowish. This is because we did not perform the purification using column chromatography. After that, the product is sampled and we determine the structure using NMR and FTIR analysis.
The impregnation of ionic liquid into the activated carbon is prepared by the incipient impregnation method at room temperature. The materials were prepared with 4 different loadings of ionic liquid onto the activated carbon. First is 0%, second is 10%, third is 5%, and fourth is 2.5%. The amount needed for the materials is shown in the slide. And then the mixture is gently mixed with methanol and stirred for 15 minutes at room temperature. Methanol is then slowly removed using the rotary evaporator that will be resulting in physical immobilization of ionic liquid into the activated. And finally, the impregnated of activated carbon is dried and stored into the desiccator. Then, the filtration process is conducted at the water filtration system at lab 200105. As attached in figure 11, that is the filtration layers that we have produced. And then the contaminated river will flow through the layers at the top of the filtration column, and then the product will flow out from the bottom of the filtration column.
After we have synthesized our ionic liquid, we sample the ionic liquid for NMR analysis in order to determine the structure of the organic compounds in our ionic liquid. NMR takes advantage of the interactions of nuclear spins in a powerful magnetic field, and therefore NMR signals are produced by unpaired nuclear spins. In our case, we have recorded the NMR spectra on Bruker at 1200 series spectrometer, and we actually ran a 1H NMR, therefore only protons will be recorded on spectra. The NMR analysis was run at 500 megahertz. The deuterated solvent used for our sample was acetone D6. Besides NMR, we have also sent a sample of our ionic liquid for FTIR analysis. FTIR is categorized and identifies the components by having molecules absorb specific IR frequencies that match the vibrational frequency of chemical bonds present in the molecular structures of our ionic liquid. In our case, we performed the FTIR analysis on Perkin-Elmer Frontal 01 spectrometer. Lastly, we have also performed AAS analysis for our project. AAS determines elements in the sample, which in our case is to measure the concentration of lead and copper ions to test the efficiency of the produced adsorbents in terms of metal removal. AAS applies characteristic wavelengths of electromagnetic radiation from light sources to excite atoms. Different elements will have different wavelengths absorbed. The amount of light absorbed is measured, therefore data will be produced as concentration of interest analyte in the sample. Using the concentration of elements obtained from AAS, we will be able to calculate the efficiency of our produced adsorbents using the formula shown in the slides.
Okay, so for the project planning, after we obtain the group, which is consists of me, Izzah, Madeline, and Rachel, and we got already our topic, we do discussion among each other and we do literature review as suggested by our supervisors, which is Dr. Cecilia and Sir Faisal. Then we do task distribution with each other, with Izzah being the group leader. Then we prepare the activated carbon, which is done by Izzah, and ionic liquid, which is done by me and Madeline. After that, we analyze the ionic liquid using NMR and FTIR, which is done by Madeline and Rachel respectively. After that, we incorporate the ionic liquid into activated carbon, which is done by Izzah. And then the sediment filter system is prepared by Rachel. And then we collect the river water source from Kinta River. And then all of us do the filtration process at block 20 and we produce 6 types of sample. And then all of us we do analysis using AS based on the 6 samples that we have done. So this is the task distribution which is distributed among our group members. So all of the tasks are distributed fairly.
Moving on to results and discussion. Starting from the NMR analysis, there were traces of impurities found in the ionic liquid, which can be attributed to the lack of the purification process after synthesis. Apart from that, 50 hydrogen atoms have been detected in the analysis. The peaks are then assigned to the different structures in the compound as per the multiplet report generated for the analysis, and with final confirmation all 15 hydrogen atoms belong to the beaming cation of our ionic liquid.
Moving on to the next part, for the FTIR analysis, we have used this analysis to get a confirmation on the structure that we have in our ionic liquid. So basically, we have detected about 6 peaks that can be used to analyze the structure of the ionic liquid that we have. So the peak is, then the peak that we found in the graph is they refer to the IR correlation table in order to identify the type of bond exists in certain frequency range. As you can see here, all of the stretch found in the FTIR graph can also be found in the expected structure in the ionic liquid, starting from the NH stretch to the CN stretch.
So this is the result we obtained from analysis of AS. So the sample names are labeled below the slide. So to conclude, all the samples concentration after filtration, which is number 2, 3, 4, 5, 6, decreases compared to before filtration, which is the number 1. Filtration using the 10% by MDCN, which is the number 5, produced the highest efficiency to remove the Cu by 11%. The recyclability, which is the number 6, is proven work as actually managed to reduce the amount of Cu compared to before filtration. However, the results are affected by errors during the experiment. For example, like contamination in the activated carbon and leakage during the filtration. As the previous slide analyzed Cu, this is the slide analyzing the STP. As we can see, all filtered samples concentration increases compared to before filtration. This is not logical as it does not follow the literature at all. So we made some inference. Maybe the river water contains very little amount of plumbum. The river water that we had does not store properly, and it is difficult for us to determine the change of concentration from very tiny little concentration. So we conclude that the results are invalid. So to overcome the previous problem that we occurred during the experiment, we rerun another analysis, but instead of using river water, we use 1 ppm of plumbum. So we run filtration using blank, 2.5% and 5%. So for 2.5%, it is higher than blank because of errors during the experiment. So after we overcome the errors, we managed to get the correct value for 5% as it produces efficiency of 55% that can filter out the plumbum. However, we cannot obtain any results for the 10% as we did not have enough sufficient amount of product to be run the experiment. So to conclude, our filtration system actually can purify Cu and plumbum.
So for this project, we have spent about 195 ringgit and 51 cents, which is for the preparation of our ionic liquid, which are BBMCl, and also sodium diacetylamide, and also our activated carbon, which is the charcoal powder. So we have spent about 123 ringgit and 33 cents for our BBMCl, 47 ringgit and 48 cents for the sodium diacetylamide, and also 24 ringgit and 40 cents for the activated carbon. So from the quantity of grain absorbed that we have successfully compared the efficiency of heavy metal removal for lead and copper by 4 different loadings of ionic liquid into activated carbon. So the highest result shown, 20% of lead and about 11% of copper can be removed from the initial sample.
We will now proceed on to the final segment of our presentation, which is conclusion and also recommendations regarding this project. As a closure, the higher the concentration of ionic liquid in activated carbon, the higher the efficiency of heavy metal removal from water during filtration. This was especially proven from the filtration run using 10% of diamine diazonamide incorporated activated carbon adsorbent, with the highest efficiency in copper metal extraction, as 11% of copper has been removed from the initial sample. A similar outcome for lead removal efficiency should also occur for 10% of diamine diazonamide incorporated activated carbon adsorbent, as deduced from the AS result obtained for the filtration performed on 1 ppm lead standards using 5% diamine diazonamide incorporated activated carbon with 20% of lead removed from initial sample. As for the recyclability of ionic liquid enhanced activated carbon, 10% of diamine diazonamide incorporated activated carbon adsorbent retains moderate efficiency in copper metal extraction after HCl rings, with 4.21% of copper removed from the initial sample. It is expected that a similar outcome for the adsorbent would be obtained for lead metal extraction. Overall, the objectives of this science team project has been achieved.
Since we only obtain very low percent of heavy metal extraction, we could actually take into consideration of some recommendation to increase the extraction efficiency. For example, first, use the high concentration ionic liquid. Mole contacted of diamine diazonamide with the aqueous phase in the extraction example is only 0.0731. So if we could get a high concentration of extractant, that may give better extraction properties. Second recommendation that we can also take into account is use the cation in ionic liquid with higher hydrophilic character. It is known that diamine cation itself has high hydrophilic character, but if we could compare with some other cations like tetraethylammonium that has very high in polarity, it may favor the mole transfer of the metal in the ionic liquid phase via the cationic exchange process. And last but not least, purification of ionic liquid could also be made before impregnation and structure analysis through column chromatography, as it may reduce traces of impurities from the compound and improve the efficiency of ionic liquid in the metal extraction. And finally, these are some of the references we have referred to in this presentation. This marks the end of our presentation. Thank you. We may now proceed with the Q&A session.